5 lectures left.
I know.
So any questions? You know where you are this morning? So what do plasma cells do?
These antibodies.
They secrete antibody, a whole bunch of antibody. Okay? What kind of signal or signals do they need to be able to kick in to start producing antibody? Their parent cell had to be stimulated by the antigen that matched their B cell receptor, right? And once that stimulus happens, then the cell starts dividing. We had clonal selection, remember? The antigen selects which of those B cells to bind to and to turn on. And then it starts differentiating and produces plasma cells and memory cells. Remember that? And the plasma cells are the ones that produce antibody. There are some types of antigens that can stimulate the parent cell on their own without any help. What do we call those kind of antigens? Not super antigens. Well, super antigens stimulate T cytotoxic cells. They stimulate T cells, not B cells. Remember T-independent antigens? And what characterizes a T-independent antigen? What kind of molecule is it? Typically molecules that have a lot of repeating subunits, like a carbohydrate or a lipid. The majority of antigens, however, are T-dependent antigens. So those cells that respond to T-dependent antigens need a minimum of 2 signals to start dividing and differentiating. They still have to find their partner antigen. But additionally, there have to be some cytokines that are produced. And typically those are produced by T helper cells. They might be produced by the antigen presenting cell. But this idea of multiple signals to get the cell going and start producing plasma cells to produce antibodies, more often than not, you need more than that one signal of binding antigen. Okay, remember that during education, cells go through this gene shuffling phenomena and eventually become locked into producing a particular type of receptor. And once you're locked in and can produce that receptor, then you're educated and you can go off to the lymph nodes and kind of wait to do your work. Okay? If you are stimulated then by your antigen, you secrete an antibody that is very, very much similar to that receptor that you bear on your surface. It actually has the same antigen specificity. Okay? But one of the things that's going to make things a little bit more confusing is that with antibodies, there are actually different classes of antibodies that we're going to have to talk about. So once you have locked in and have a particular receptor, you will then start secreting an antibody, if you get the proper stimulus, that has the same antigen specificity as that receptor on the cell that produced the antibody. But the antibodies can belong to 5 different classes. All the classes still have the same antigen specificity, but the different classes of antibodies have little different properties in terms of some of the kind of general things they can do. So that's something we need to dig into a little bit in just a minute. Okay, so here's kind of a generalized representation of what an antibody might look like, right up here. And this looks a whole lot like a B cell receptor. Okay? two heavy chains, two light chains, a constant region down here, variable regions here, okay? Antigen binding specificity here and here. Same molecule of antigen binds here as binds here. So this is one of the types of antibody molecules that activated cells can start cranking out and producing. And this particular antibody belongs to a class called IgG antibody. There are 4 other classes of antibody that can be produced also. And those are classes A, E, M, and D. How are all of these similar to one another? For a given cell that has a given receptor, if you can produce all of these different classes of antibody, they will all react with the same antigen. So this region right up here at the top, the binding regions for antigen will be identical, whether you're an A, an E, a D, an M, etc. Okay? What differs this particular region down here? This particular region called the FC region, mainly made up of these 2 heavy chains with that constant region of genes that encode for it. The nature of your FC region defines what class of antibody you belong to. Okay? So a class A, this would be the same as a class G, but this would be different down here. For a class M compared to a class G. These regions would be the same. They bind the same antigen. But this region down here, this FC region, would be different. Before we get into the classes, I'd like to talk about antibodies in general. And specifically, what is the importance of secreting antibody? Why is secreting antibody protective? Why is it an important part of your immune reaction? Well, we've already talked about one of the functions that antibodies can have that are protective. And that is, if an antibody binds to an antigen on a pathogen cell surface, then once that antibody attaches, it can fix complement and initiate the complement cascade. And remember what that cascade terminates in? That attack complexes, which results in holes being punched basically in the membrane of the cell. And on the way to producing that attack complex, remember that all of those cascade reactions can produce things that stimulate inflammation, for example, which help to augment the process of getting rid of this pathogen. So fixing complement is one of the important roles that certain classes of antibodies can play. What determines whether you can fix complement? This region down here at the bottom. That's why this is designated as FC. This is the region that in some classes of antibodies fixes complement. Okay? Not all classes of antibodies can do that. We'll show you a comparative chart in a minute. Some classes of antibodies fix complement, others don't. What are some other reasons to produce antibodies? Okay? If antibodies bind to the surface of a pathogen, then this FC region on some of these antibodies right here can bind to receptors on phagocytic cells. That's going to enhance the possibility now that the antibody, which is bound to the pathogen, will now get phagocytized by the white blood cell. Because the antibody is sandwiching, basically, the white blood cell and the phagocytic cell, and the pathogen, sorry. So the antibody is kind of a bridge. It joins the pathogen, it joins the phagocytic cell, and when they're bound together, it's more likely that that pathogen will be internalized. So that's another role in some classes of antibodies that this region, this little stem down here plays. It binds to phagocytic cells. Antibodies that enhance phagocytosis are called opsonins. Okay? So that's another way in which antibodies can help to facilitate destruction of pathogens is by being opsonins. There's another phenomenon that can take place. Antibodies can bind to surface molecules on virus and cover up the receptors on the virus particle. And what is the first thing that has to happen before a virus can infect the cell? It's got to attach. If it does not attach, then it cannot infect. So these are sometimes referred to as neutralizing antibodies, okay? They bind to the virus and cover up their attachment sites, therefore they can't bind to cells and infect them. Another thing that antibodies can do is that because antibodies have at least 2 binding sites for the same antigen, and we're going to see later on some have more than 2 binding sites, they can cross-link antigens on different cell surfaces. So this antigen binding site here can bind to this cell, and on the same antibody, this antigen binding site can bind to this cell. This phenomena of cells getting grouped together in kind of a lattice is called agglutination. And if you start forming these agglutinated clumps, it's more easy for a phagocytic cell to engulf those clumps than it is individual bacterial cells. Something very similar happens with antibodies that bind small soluble antigens. In this case, not cell surfaces, but small molecules like toxin. And they can cause a precipitation reaction by cross-linking these toxin molecules, again, enhancing their phagocytosis. So the big difference between agglutination and precipitation is here the antigens are part of cell surfaces, here they are individual molecules like toxin molecules. And we're going to hear these terms again later on. The week after break, we're going to talk about using antibodies for testing, and we will talk about agglutination and precipitation reactions in the antibody testing section. Okay. Not sure if it's up here. No, didn't have it up here. Another thing that can happen some antibodies, when they bind to a cell surface or bind to another molecule, have enzymatic activity. And so the antibody actually gets activated to become an enzyme. And if that antibody is hooking up to a macrophage, remember we said that stem region, that FC region, can bind to the surface of a macrophage. That can activate that macrophage to produce some killing mechanisms. Like maybe secreting hypochlorite. Or producing a lot of peroxide. The antibody binding to that macrophage actually stimulates those killing abilities of that macrophage cell. This is called antibody-dependent cell cytotoxicity. So there's a lot of important things that antibodies do to eliminate the pathogen. And the amount of antibody that can be produced by an activated plasma cell is really, really impressive. I showed you that picture of the plasma cell and all of the endoplasmic reticulum that it was filled up with. Those cells really become devoted to primarily one function, and that is transcribing those genes for antibody production, packaging them up, shipping them out of the cell, and secreting them into the environment. They burn out in probably 5 to 7 days. They're producing thousands of antibody molecules per minute. And that really just becomes kind of the reason for existence for these plasma cells. Is don't worry about anything else. Just start producing as much antibody as you possibly can. And that starts flooding the tissues, it gets into the circulatory system, and then it helps to activate other aspects of the immune system. Turn on the complement system, enhance phagocytosis, stop virus from binding to other cells, stop the infection. Okay, so here are our 5 classes of antibodies. If the antigen is a T-independent antigen, one of those ones with lots of repeating subunits, a polymer like a carbohydrate, a lipid or something like that, remember that these molecules can bind to, because they have the same epitope over and over again, can bind to multiple receptors, and that causes those receptors to cluster, and eventually that causes endocytosis to occur. And that's basically the signal then that's needed to cause this cell to start dividing and differentiating. T-independent antigens, so, only produce plasma cells. They do not produce memory cells. And the type of antibody that they produce is an antibody called IgM. Okay, take a look up here at the different classes of antibodies. And here's a visual representation of each of the types of antibodies. This compares their number of binding sites, their molecular weight, percentage of total antibody in serum, average life in serum, and then some interesting properties that members of that particular class of antibody has. We call these kind of general properties that any antibody within that class can do. We call those effector functions. So take a look at what some of those effector functions are. Can it cross the placenta? That's an effector function. Can it fix complement? Can it bind to phagocytic cells? And as you start looking through these, you'll see that some classes of antibodies can do some of those things, and other classes cannot. So the effector function kind of defines general activity of an antibody class. And that's going to be really important in terms of dealing with pathogens entering from different sources in the body, for example. Okay. So, here's the one we use as kind of our general model of antibody structure, IgG. As it turns out, this isn't the first antibody that's produced. But it's a simple structure, so that's kind of usually the one that's used for illustrative purposes. So let's look back at IgG again for a second, okay? So number of antigen binding sites, 1, 2. It has a valence of 2. Don't worry about understanding or memorizing these molecular weights right now. One of the things probably that might be of use understanding is that IgG is the primary antibody that you find in serum, in the blood. This is the primary antibody. Okay. Another thing that's probably important to know is this is the one that tends to have the longest lifetime. It has a variety of effector functions, okay? It can cross the placenta, it can fix complement, and it can bind to phagocytic cells. It's a pretty versatile antibody. This particular antibody, IgG, is the one that really is involved particularly in the memory response. And we'll dig into that just a little bit later on. Okay, contrast that with IgM. Here's IgM. That's a big old hunk of antibody, okay? Look at its molecular weight, 900,000 versus 150,000. It's composed of basically 5 of these IgG molecules held together by a polypeptide chain called the J chain. So if it's 5 IgG-like molecules, it has a valence of 10. Do you find this in the serum very much? No, actually you don't. Even though this is the first antibody that is typically produced. Later on, it's replaced by IgG. It's got a pretty short lifetime. It can fix complement. It won't bind to phagocytic cells. Why won't it cross the placenta? It's too big. So this is really the first antibody that is produced in response to an antigen. And it's the only antibody that is produced in response to a T-independent antigen. So when you have a response to a T-independent antigen, it's a pretty short-lived response. The antibodies don't really last that long. What if the antigen is T-dependent instead? Okay. So here's a cell. That is going to attach to an T-independent antigen. Okay, so here's an antigen. It's actually going to be part of a cell. Pretty pathetic cell. Here's a cell with antigens on its surface. It binds to these. But what we're going to see is that in order for this response to occur, this cell has also got to interact with the T helper cell. This binding of the antigen to the cell is one signal. The T helper cell, when it binds, produces cytokines, which provide the second signal. So now when this has gotten its 2 signals, it starts to divide. Okay? And it produces plasma cells. But it also produces memory cells. Memory cells have receptors still on their surface. Plasma cells do not. These plasma cells then start cranking out antibody. IgM first. And this particular antibody has the same specificity for antigen that this receptor on this parent cell had. But what can and usually does happen is eventually this plasma cell starts producing a different class of antibody. We call that phenomena class switching. And it starts producing an IgG antibody. Memory cells, if they attach to their partner antigen, can immediately start producing IgG. They don't have to go through and produce M first and then switch classes. They can immediately start producing IgG. Some cells can further switch class, if they get the right signals, and maybe even produce IgA or IgE. So what's involved in class switching? Remember, to make an antibody, you need to make heavy and light chains, right? And heavy chains and light chains both have variable regions and constant regions. Those cells, when they built a receptor, locked in those VDJC genes. And once they're locked in, they always have the same receptor. And when they're stimulated then by the antigen, they transcribe those same gene blocks and produce antibody instead. They never change those V, D, and J blocks. But in class switching, they can splice a new C region onto the VDJ. And that is what produces the new class of antibody. So that's what's involved in this class switching phenomenon. Well, this helps to explain why immunization works. Because this initial activation of a cell to become a plasma cell, to produce IgM, takes a while to get going. This differentiation of a memory cell into a secreting cell doesn't take much time at all. And so when you immunize, you're trying to basically build up a population of memory cells. And then if you see that antigen for real, then primarily what you're doing is stimulating memory cells, which start cranking out IgG very quickly. I was going to show this, but I'll go ahead and show you this right now. So here's a typical kind of kinetics of an immune response. We break the immune response down into primary response and secondary response. The secondary response is referred to as the anamnestic response or memory response. Okay? Primary response is the first exposure to antigen. First time your immune system's ever seen it. Okay? So that involves that educated lymphocyte having its receptors bound to the antigen for the first time. So that then causes this differentiation into plasma and memory cells. We start producing IgM first. So we start seeing a spike in IgM. And then eventually they can class switch and start producing IgG. So here's our total antibody production. Here's the antibody production broken down by class. Okay? Threats eliminated. Or this was an immunogen that doesn't cause disease. But now we've got these plasma, these memory cells. If they are then stimulated by their antigen, because remember, they still have a receptor, so they can recognize antigen, they immediately start cranking out IgG. So IgG really spikes very quickly. And there's not this lag period. There's not this latent period. Because these cells have already gone through a partial differentiation process. These are primed and ready to go. Okay? This initial stimulation takes some time. That's the latent period. Now, could there be some new naive cells that have never seen the antigen before that are also stimulated this time? Sure. And what would they produce? They would produce IgM. But there would be that lag period because it would take them time to get geared up. So if you look at the memory response, total antibody production is much higher. IgG production is much higher and much earlier because of that memory response that's occurring. Okay, what do some of these other classes of antibodies do? IgD, we're not really sure. Probably more likely than not, this is just kind of the receptor that is on that educated cell. And so you can kind of think of IgD as kind of the B cell receptor. IgE is involved in allergic reactions and protection against parasitic worms. If you suffer from allergies, what's really happening is that when your lymphocytes start getting stimulated by those allergens, it activates plasma cells, and they very quickly just switch over and start producing IgE. And they way overproduce IgE. And IgE is interesting. Its stem, its Fc region, binds to mast cells and basophils. And when it binds to those cells, it causes them to degranulate. And when they degranulate, they release things like histamines. What do histamines do? Inflammation. Okay? And so basically, in response to these allergens, through the overproduction of IgE, you start getting an inflammatory reaction. And many of the symptoms of allergies are the same symptoms of inflammation. IgA is an interesting class of antibody and a very important class of antibody. Okay, let's take a look at it for a second. How many binding sites does it have? Well, it depends upon the type of IgA. There's two forms. There is serum IgA and there is secretory IgA. Serum looks a lot like IgG or D. Okay. Secretory is two of those molecules put together by a secretory component. And this allows the antibodies to be shipped out of tissues into the external environment. IgA is very important in helping to protect against pathogens at common portals of entry like the respiratory tract and the digestive tract. When those lymphocytes that are associated with the GALT, the gut-associated lymphoid tissue, the MALT, the mucosal-associated lymphoid tissue, when they get stimulated, they start producing that secretory IgA. And the cells lining the intestines, for example, or breast tissue in a lactating mother, actually stick this secretory piece onto the IgAs, and that allows those molecules to become transported across the membrane and secreted. Well, think about it. If you're now basically secreting IgA onto the surface of your gut or the surface of your respiratory tract, then if you inhale pathogens, you're going to have that protection before it even gains entrance. So that gives you a first line of defense against these pathogens. If you're a nursing mother and you start secreting IgA into breast milk, then that gives the infant protection. Where did the protection come from prior to birth? It came from IgA, for example, which can cross the placenta and give the fetus immunity, the same immunity the mother has. Okay? But once that infant's born, you lose that immunity to IgG. Okay? There's no placenta anymore. If you did not have the IgA secretory in the breast milk, then these infants are basically relying on just their own immune system. And yeah, they start developing an immune system pretty early on. As they start picking up their normal flora, it takes time for an immune system to develop. You know, kids get sick more often than adults do. They've got an immature immune system. But if you can give that infant some temporary protection due to secretory IgA, it really does help to augment their immune system. And there's lots of epidemiological studies that say kids that are breastfed have fewer infections than kids that are bottle fed, because bottle feeding does not give you that protection of IgA. When we look at immunity, there are two major types of immunity. There's natural immunity and there's artificial immunity. Natural immunity is achieved by actual exposure to a pathogen or a toxin. Your immune system's being challenged by the real deal. Okay? Artificially is produced through medical procedures, including immunization. Okay? In both natural and artificial immunity, there are subsets. There is active and there's passive. Okay? So let's look at natural immunity for a second. What's the difference between active immunity and passive immunity? Active is a consequence of a person actually developing their own immune response and secreting their own antibodies. So if you get mumps, for example, you develop an immune response against that particular pathogen. And remember that there is a lag period involved in that primary immune response. And likely the response is not going to occur fast enough or robustly enough that you'll get symptoms associated with that particular infection. But hopefully you will recover, and then you will have memory. You will have a natural active immunity. Passive active involves getting antibodies from another source, in this case, your mother. Remember that antibodies have a limited lifetime. Okay? Remember average lifetime? G, 23 days. A, M, all of these just a few days. So the antibodies you get from your mother aren't going to last forever. But there is no lag period. You're getting the actual antibodies. And so as soon as they are introduced into your body, they can start responding to antigen. So no lag period here, but protection is temporary. And once you discontinue the source of antibody, the breastfeeding, then that protection stops.
When I was looking at the Ebola stuff, where the procedures that they use is like blood transfusions that already have antibodies for Ebola. Would that be the same?
That will be artificial passive.
Oh.
Okay, which is where we're going next. Okay? So remember, this happens through natural phenomena. Exposure to antigen, the real antigen, or breastfeeding. Artificial immunity is medically induced. Okay? Artificial active involves being exposed to a weakened form of the pathogen. The idea is that we treat it so that it maintains its antigenicity, its ability to stimulate cells, but its ability to cause disease has been compromised. One of the easiest ways to treat these particular antigens is mild heat treatment. You kind of partially denature it, it's still antigenic, but it doesn't cause disease. There are various chemical treatments that you can use to do this as well. They used to use formaldehyde, for example, to modify the natural antigens and then use it in immunization. So now when we are exposing someone to these artificial substances, basically, these weakened antigens, they do stimulate the immune response. And you go through that lag of the primary response, you start producing IgM and IgG, but you produce memory. And in subsequent exposures, we'll be stimulating a lot of those memory cells. Secondary response now, no lag. And so oftentimes, usually, you're producing enough antibody that you can fight off the infection before it ever really causes any overt symptoms. Passive immunity is the consequence of receiving antibodies from someone other than your mother through breastfeeding. So that could be the blood of a person who has survived a particular disease, like Ebola. That person has mounted their own immune response and are producing antibodies. Sometimes you will take blood from several different sources of individuals and isolate out just the globulin, the gamma globulin portion of the blood, which is the antibodies, and then you can get infusions of those pooled gamma globulins from a number of different individuals. That's not an uncommon response to an outbreak of something like hepatitis. Hepatitis is often a foodborne disease, and if there has been a restaurant, for example, that epidemiologists have traced back to a hepatitis infection, then they'll send a notice out, and if you have eaten at that restaurant within a defined period of time, they'll say, go to your doctor, and the doctor will very likely say, we're going to give you some protective gamma globulin. Okay? And that shot will contain antibodies from people who have contracted hepatitis and have survived and have produced their own active immunity against it, and therefore are secreting antibodies. And those give you some temporary protection while your own immune response is gearing up, if indeed you have been exposed. Okay? Which one gives you a more robust and long-lasting immune response? Active, natural, or active passive? Active natural, yeah. Typically, exposure to the real deal gives you kind of lifetime protection. But the immunization sometimes will require boosters because the immunity that you get from that artificial active is not as long-lasting. Okay, so we're finally ready to kind of put things together now. So what we're going to do next time is we're going to talk about the two branches of the adaptive immune system, the line 3 system. And those are the cell-mediated immunity and the humoral immunity. We're going to start talking out first about cell-mediated immunity, and then that will lead into humoral immunity. The major players here are going to be B lymphocytes, T helper cells, and antigen-presenting cells. And different cells react with one another in the humoral and cell-mediated immunity. It's very important to understand that binding of antigen to a receptor is one of the signals that has to happen in order for these types of immunity to be activated. More often than not, we need additional signals in the form of cytokines. And that's where those helper cells will really come into play, is producing some of these cytokines that really provides the additional signals we need to make this happen. So that's what we'll do Tuesday. 5 minute break. I'm still here.